Hello everyone, my name is Labarge and I recently completed my PhD with the University of Buffalo. And this research comes from my dissertation where I investigated the spatial and physiological aspects of the ecology of fear in a wild primate. In this study, I examined whether human presence and other potential ecological challenges may be associated with variation in fecal cortisol metabolites in wild samango monkeys. Samangos are also known as sykes or blue monkeys, and my particular study population in South Africa's Soutpansberg Mountains experiences some mild seasonal variation in resources, rainfall, and overnight cold temperatures, and are subject to potentially dangerous intergroup competition and predation. Yet one complication to looking at the effects of predation or competition risk is that previous research at our site indicates that our subjects experience a human shield effect, where observer presence reduces the likelihood of encountering an unhabituated predator. This may also apply to unhabituated competitors as well and affect animals' perception of how dangerous their environment is. Alternatively, our presence may also affect these animals because observation itself may be stressful even after animals are considered habituated. Here I focus on collecting repeated samples over time from known individuals in a single habituated group to measure changes in baseline levels of fecal cortisol metabolites, or FCMs. This study group spent 3 to 4 days each week unobserved by researchers, and because previous physiological validations indicate a 23 to 25-hour lag between an event and changes in FCM levels, that meant that we could evaluate whether cycles of human presence or absence might help predict variation in FCMs through reducing perceived risk or by observation acting as an additional stressor. Additionally, using a subset of data where we did have information from 24 hours prior, I also investigated whether the cumulative number of predator or intergroup encounters affected FCM levels the following day, and whether increasing numbers of observers in the field might modulate responses to these events through a human shield. Now, while single events are too ephemeral to detect with changes in fecal metabolites, risk perception throughout the day may be important for baseline FCM levels. I initially conducted an exploratory analysis looking at the importance of ecological variables like food and water availability, temperature, and human presence, and compared support for these candidate models, as these could all plausibly serve as physiological or psychological stressors. I then used a subset of data where I had information on the previous day to test specific predictions about the effects of predators, competitors, and observer numbers. So specifically, I predicted that as the number of predator or intergroup encounter events increased per day, then baseline levels of FCMs would be higher the next day. I also predicted that as the number of observers with a group would modulate these responses due to a human shield. But alternatively, if these animals are still sensitive to human presence, then increasing numbers of observers might then be associated with a rise in FCMs. We collected fecal samples from 13 known adult females in a group of around 75 monkeys, and we conducted field extractions and then after transport stored samples at -20 degrees C in the US. We limited our collection to the early morning period to minimize the effects of circadian rhythms and aimed to sample evenly between days when animals had been with and without an observer the previous day. During the day, we also conducted behavioral observations where we recorded all alarm calls, which tend to only happen in response to predators, and we also recorded all intergroup encounters. The most common predators encountered on our site were black and crowned eagles, although samangos also alarm call at leopards and snakes as well. We collected temperature and rainfall data on site with missing values filled in with the Copernicus Climate Data Store, and these satellite-derived data were strongly correlated with our on-site measurements. To measure food availability, we collected data on the phenology patterns of 26 food tree species from randomly spaced vegetation plots and calculated an availability index for each month adjusted for the importance of each species. At the end of sample collection, I obtained 192 usable samples with an average of 14 per individual. I validated and conducted enzyme immunoassays using commercially prepared kits and conducted these at Canisius College between October 2019 and March 2020. We used a model comparison approach to evaluate the importance of food and water availability, thermoregulation, and cycles of observer presence. Daily variables were set on a 24-hour lag to match the time it would take for FCM levels to change. In each model, we also controlled for energetically demanding things like infant carrying status, and we included individual ID as random intercepts. We also compared versions of each model with and without random slopes to allow for variation between individuals, so where individuals might have diverging responses to the same stimuli. We performed Leibniz cross-validation to calculate predictive densities for these models, and then used model stacking to compare weights. Following this initial analysis, I asked if external events of intergroup encounters or predator encounters might be associated with changes in FCM levels, and if such physiological responses varied with the number of human observers present throughout the day. Here I used a subset of data for which I had information from the previous day and set the previous day's cumulative incidence of each encounter type as fixed effects in a single model. I used a count of the number of observers as an interaction with each encounter type. In my initial exploratory analysis, stacking weights were shared nearly evenly between models for resource availability, thermoregulation, and rainfall. But my models incorporating information on human presence had little support, and so did models incorporating random slopes. Specifically, higher seed availability was an important predictor of lower FCM levels. Lower minimum temperatures during the day were also associated with higher FCM levels, and lower total rainfall per week was associated with higher FCM values, but only in the dry season. I found with my subsequent analysis that with only a single observer present, a greater number of predator encounters observed during the day was associated with higher FCM levels. However, with 3 or more observers present, the apparent positive trend flattened. In contrast, I found no such relationship for intergroup encounters. Water scarcity, food availability, and thermoregulation appear to have important physiological effects on this population, but unexpectedly I found no evidence supporting the idea that human researcher presence or absence alone might affect variation in samango FCM levels. As the number of alarm calls that occurred throughout the day increased, so did measured FCMs the next day. Responses to potential predator encounters, though, were apparently dampened when 3 or more observers were present. Multiple observers may be more likely to inadvertently deter unhabituated predators, such that the stimuli that trigger widespread alarm calling may tend to be less serious than those that occur with single observers present. It might also be that in large groups, individuals are unlikely to be near a single observer for long periods of the day, so that as observer numbers increase, a human shield effect might be perceived more consistently across individuals. Unfortunately, our study group inhabited such a dense environment and group spread was so wide that this restricted our ability to record the cause of many alarm calls. So I don't really know for certain whether this result is due to a reduction in the riskiness of encounters or due to a reduction in risk perception or both of these things. So thank you so much for listening to my presentation, and if Wi-Fi allows, I will be here to answer any questions.